In this video, you'll find a demonstration of how to use CF11 columns to remove white blood cells, or leukocytes, from samples of malaria-infected whole blood. Depletion of leukocytes reduces the amount of human DNA and RNA in the sample, improving the quality of parasite genomic and transcriptional analysis. You will also learn how to store samples for DNA and RNA isolation after passing the blood sample through CF11 columns. Safety precautions for handling blood and infectious materials must be taken at all times during this procedure. These include the use of protective eyewear, shoes, and clothing, the use of gloves at all times, and proper waste management. For the track study, 10 milliliters of venous blood will be collected from adults for DNA and RNA preservation. In children, we will collect 5 milliliters. Collect whole blood samples in blood tubes containing EDTA. Do not use heparin, which can inhibit Taq polymerase. Label sample collection tubes with study subject number, study subject initials, and date and time of sample collection. Invert gently to mix well. Store blood samples at 4 degrees Celsius until they can be processed to remove leukocytes. Proceed to leukocyte depletion as soon as possible, within 6 hours of sample collection. Leaving samples at room temperature or freezing samples prior to leukocyte depletion will result in hemolysis and significant sample loss. Leukocyte depletion requires CF11 columns, isotonic PBS or rPMI buffers, 15 and 50 milliliter centrifuge tubes, serological or transfer pipettes, bleach solution for waste disposal, sticky tape, and a centrifuge. One column should be used for up to 5 milliliters of blood. Blood volumes higher than 5 milliliters should be split among multiple columns. Gently remove the plunger from the column and make sure that the column remains intact. If cellulose powder gets displaced by air, push the plunger back in to secure the column again and gently remove. Suspend the column over an uncapped, labeled 50 milliliter tube using a clamp or tape. Apply 6 milliliters of PBS to the top of the column. While PBS passes through the column, prepare your blood sample. Transfer blood to a 15 milliliter centrifuge tube. If the sample is being split among multiple columns, split the sample into multiple 15 milliliter tubes. Centrifuge at 1000 G or RCF for 5 to 10 minutes. You should see a red blood cell pellet and a clear separation of pellet from supernatant. Carefully remove and discard as much of the clear supernatant as possible. A small amount of red blood cells may also be lost. Add PBS to the blood so that the total volume equals twice the original blood volume. For example, for a blood sample with original volume of 5 milliliters, add PBS to the pellet to total 10 milliliters. Gently pipette or invert to mix. Make sure that the PBS has passed through the top of the column. Then apply the blood PBS mixture. Depending on volume, you may need to apply the sample in 2 steps to prevent overflow in the syringe. Allow the sample to pass through by gravity. Once all of the sample has passed through the top of the column, add 6 milliliters of PBS. Add the PBS within 10 minutes of the sample no longer being visible at the top of the column. Otherwise, the column will dry out, causing loss of sample. Allow PBS to pass through until there is no fluid remaining at the top of the column. This will take approximately 10 minutes. Carefully reinsert the plunger and gently push to pass the remaining sample through the column in a dropwise fashion. Once the plunger has been inserted, you cannot add any more sample or PBS to the column, because removal of the plunger will allow air to disrupt the column. Centrifuge the filtered sample at maximum speed at room temperature for 5 to 10 minutes. You will see the red blood cells pellet and a clear separation of pellet from supernatant. Discard the supernatant, taking care to not disrupt the red blood cell pellet. It is preferable to leave a small amount of supernatant, up to 1 milliliter, rather than disrupting the pellet, because this may result in sample loss. Storage of packed red cells for DNA and RNA requires trizol reagent stored at 4 degrees Celsius, pipette tips and a micropipette, transfer pipettes, and cryovials for RNA and DNA storage. For RNA, transfer 500 microliters of leukocyte depleted packed red blood cells into a cryovial labeled with study subject number, subject initials, and date and time of sample collection. When using Trizol reagent, be sure to use protective eyewear, clothing, and gloves. Avoid inhalation and contact with skin, and use a fume hood if available. Add 5 milliliters of Trizol reagent to the tube, and gently pipette up and down to mix. Note that in children, where only 5 milliliters total of whole blood is taken, 300 microliters of blood and 3 milliliters of Trizol reagent will be used. After mixing, leave the tube at room temperature for 20 minutes. For DNA storage, transfer the remaining leukocyte depleted packed red blood cells to a cryotube labeled with study subject number, study subject initials, and date and time of sample collection. Store both tubes in liquid nitrogen until extraction of RNA and DNA. Pellets should remain frozen during shipping and long-term storage.